Dear listeners, I'm Dr. Charles Rwanda, lecturer from University of Rwanda. I'm here to present to you the findings of the project entitled The Prevalence and Antimicrobial Susceptibility Profiles of E. coli Isolated from Human and Broiler Chicken Feces in Selected Regions of Rwanda. The authors of the project are Charles Rwanda, Marie-Christine Dusingize, Emmanuel Dimaso, Marie-Aurore Ugirabe. The PI of the project is Marie-Aurore Ugirabe. The project was funded in collaboration with University of Rwanda and the Swedish International Development Cooperation Agency. Here again, we see the PI, Miss Aurore, and she was interviewing a farmer here. Here we see the images, different images. We have Miss Christine. She was also interviewing the farmer, asking about poultry farming things. We see the second image here. It is me who was collecting poultry feces, and I collected them from different angles of this house and pulled them together and put them in a container. The third image on this slide is just Dr. Irimas and I, and we were just analyzing fecal samples in my microbiology lab. The last image on this slide, here these are the plates, these are antibiogram results. We see discs, and around them we see the zone of inhibition. So around these discs we see the zones of inhibition, and again we use the ruler to measure the diameter of the zone of inhibition. Coming to this slide, we look at the objectives of the project and the approaches we used. The main objective was to determine the prevalence and antimicrobial susceptibility profiles of E. coli from both humans and chicken feces. Remember, humans, here we worked with people who were in contact with chicken. Method is used for identification purpose. We used bacterial culture media, especially TBX and maconche. We used the microscope, and also we used the biochemical tests, and all these confirmed the presence of E. coli. Antimicrobial susceptibility testing was conducted by Kibe blower disc diffusion method using antimicrobials commonly used in veterinary and human medicine, and these include amoxicillin, tetracycline, doxycycline, erythromycin, ciprofloxacin, norfloxacin, kanamycin, and also amikacin. The results or the findings of the project. In total, we found 309 E. coli isolates, and we obtained from humans and from chickens. Coming to the resistance, we found that microorganisms were more resistant to tetracycline, followed by amoxicillin, and the last was erythromycin. We found that many antimicrobials were sensitive, and these include ciprofloxacin, norfloxacin, amikacin, kanamycin, and doxycycline. We also found that 275 E. coli isolates were resistant to at least one antibiotic. We found that 141 isolates were multi-drug resistant. That means they were resistant against at least 3 classes of antibiotics. Further investigation is needed to characterize at molecular level the antimicrobial resistance genes and genotypes of obtained isolates. Before I wind up, We acknowledge the management of University of Rwanda for providing necessary infrastructure to carry out this work. We also acknowledge Rwanda National Reference Laboratory for providing reference E.Coli. Our gratitude goes to the UR CIDA program for financial resources for the research project. So thank you so much.